we can do that. Okay, so I will start by giving the lecture part, this is the first time we've given this course, so we'll see how it goes. I think it will. There's so much to talk about. So rather than cramming lots of slides full, I just put sort of the topics on the slides because I don't know through questions and stuff where we're going to end up concentrating on. So I think the title, though, that I put up here is really kind of fundamentally what we're talking about is from good to great, and it's really an issue in biologic drug discovery because biologics can come out of the initial discovery process really, really good, and sometimes there's pressure to just move on with them because they're good enough. And if you look at the competition down at the other end when they actually are getting into the clinic, they're not going to be good enough. But they might be good enough according to the original kind of tentative ideas of what you wanted. Your profile was something that has certain characters, minimum characters, and it meets all those minima, but it's not going to be a successful drug unless it's great. And it has to be great in many ways. And some of those you can't optimize. You have to pick the right ones. And the rest of the things you can optimize. And if you do one optimization after another, you'll end up with a fantastic molecule 5 years from now, and they will drop the project. So unless you're an academic lab with one person working on it, in which case you do that. And there isn't the time pressure, and you just triage and figure out which order to do everything in. So, not everything I say will be necessarily exactly the way you would do it if you're not in the kind of industry I'm in. But it's just really important to have an understanding that we need to improve our molecules till they can really compete and outcompete others in the marketplace and differentiate. And there might be ways of optimizing for differentiation. So, so the drug world is really unforgiving, and it won't let you get away with a molecule that's only okay. And one of the problems is competition, because I don't know what indications everyone's working on, but the big diseases have so much competition, and even down to which target. Many companies working on EGFR, many companies working on IL-6, many companies working on all these big targets. So you need a molecule that's going to be better than theirs, especially if you're not going to be the first. The other is the regulatory and commercial things. You need a molecule that will get through FDA, which is a very different animal than getting through the committees that meet to decide whether the molecule's good enough to go into the next stage of development. And people in the company at the more downstream end may know more of that than the discovery protein engineer people do. But that's very important information to try to get, is feedback of whether your molecule will be able to pass FDA. Right? But it's something, the earlier you think about that, the better for optimization, because so many molecules end up, at some point they have fixed because you're going to start process development and such. And then there are problems that you have to go way back. So where it's protein engineer looks and says, if I'd only known that was a problem, I could have fixed it a year ago, and we wouldn't have this issue. So one of the things that I hope I'll take home today is that, that early